Myself, Dipti Naik, Assistant Professor at Government College of Arts, Science and Commerce, KPM. This session is for first year Bachelor of Science students, subject Botany, and for semester 1. Okay. So, paper code is BOG 101, paper title Environmental Biotechnology. Title of the unit is Xenobiotic Compounds. Module name Bioremediation of Xenobiotic in Environment, Degradative Plasmid. Module number 29. Outline of this module is first introduction of plasmids, then types of bioaugmentation and examples of plasmid mediated bioaugmentation. Learning outcome. Once we finish this module, you will be able to answer or define plasmids and degradative plasmids, explain the methods of bioaugmentation. Introduction. Plasmid is usually circular and double-stranded segment of a DNA that is found in the cytoplasm, separated from the main bacterial chromosome. Plasmid can perform various functions, such as it can provide antibiotic resistance, helps in virulence, can perform very special type of metabolism, and it is also involved in replication of nucleic acid. Now, classification of plasmid. Okay, so there are three types of plasmids. First is degradative plasmid, virulence plasmid, and the fertility plasmids. So, degradative plasmids. These are carrying genes that confers on the host bacteria having ability to degrade organic compounds. Example, toll plasmid of Pseudomonas putida. Okay. Second is virulence plasmid, where contains Vir gene which turns the bacterium into the pathogen. Example, Ti and Ri plasmids. Fertility plasmid or F plasmids. They contain transgene that are capable to conjugate and result in the expression of sex pili. Example, F plasmid of E. coli. So in this we are going to study only the degradative plasmid. For degradation of this degradative plasmid, there are special method known as augmentation. Okay, so augmentation means here the microbes are used to inoculate contaminated soil. In this augmentation, there is a further classification: cell bioaugmentation and plasmid mediated bioaugmentation. Cell bioaugmentation means it is based on the survival and catabolic activity of inoculated microbial strains to perform degradation. Whereas in plasmid mediated bioaugmentation, technology in which donor bacteria harboring self-transmissible catabolic plasmid are introduced into the soil matrix to enhance the rate of contaminant degradation. In both this method, plasmid mediated bioaugmentation is used more, okay, as it is easier and flexible. Okay, so there are some of the factors which are affecting the efficiency of plasmid mediated bioaugmentation: biotic factor and the abiotic factor. Biotic factors such as morphological, physiological and biological characteristics of donor cell. Okay, then phylogenetic distance between donor and the recipient cell, ratio of donor and recipient cell, transfer efficiency of the plasmids, and expression of transfer gene. Now, abiotic factors: moisture, temperature, pH, organic matter, and properties that contaminate bioavailability. Now, behavior of degradative plasmids are depend on replication, efficient transfer of required plasmid from donor bacteria to the recipient bacteria. Second is the stability. Stability of recipient cell to properly express the genes. Third is the host range, whether the plasmid can self-transfer themselves with the stably replicate is a seen. Last, ability of recipient cell to properly express the genes. So now we are going to study the examples of this plasmid mediated bioaugmentation. Okay, so here first example, genes from Orthobacter species in Pseudomonas putida PAW340 are used for degradation of soil slurry. And after inoculation of this recombinant, that is Orthobacter species and Pseudomonas putida species, it was found that there was a higher degradation of chlorobenzoic acid was observed. Okay. Next example, Pseudomonas putida and E. coli. Okay. Cells harboring plasmid PJP4 can stimulate the degradation of 2,4-D in soil without substantial impact on the soil microbiology. Now, these are the references. Thank you.